Okay, so hello everyone, and again, a new topic, the final one for the metabolism of carbohydrates. As we say, the last but not the least, and this is gonna be the story on one anabolic pathway, the biosynthetic pathway of novel molecules of glucose, which is marked as gluconeogenesis. So we all know that glucose is a very important source of energy for all cells in human organism. And this energy and the sufficient amounts of glucose for degradation are obtained by those catabolic processes, starting from degradation of complex carbs, then degradation of glucose to pyruvate, from pyruvate to acetyl-coenzyme A, and degradation to carbon dioxide and water in Krebs cycle. All of those pathways producing ATP. Also, we discussed about the glycogen metabolism, which serves as the major reservoir of glucose for our body for a maintenance of blood glucose levels as homeostasis. But let's move on to something that we have mentioned from time to time in attempts to explain some other metabolic pathways, and that is gluconeogenesis. And I think that we have mentioned a couple of times that in the case of absence of carbohydrates in the diet or during prolonged fasting, the amounts of glucose which are required for the maintenance of normal metabolism are obtained from the process of glycolysis within 10 to 18, maybe 20 hours top from glycogenolysis. But during prolonged fasting or even starvation, glucose is synthesized from other precursor, and that pathway of resynthesis of glucose from other non-carbohydrate precursors is explained by the pathway gluconeogenesis. Actually, if we want to locate the gluconeogenic pathway, it mainly and mostly occurs in liver. So liver gluconeogenesis actually provides approximately 90% of newly resynthesized glucose, and other tissue and other organ which is also involved in gluconeogenic pathway is kidney cortex, but it provides only up to 10% of a newly synthesized glucose, but that glucose is predominantly used for kidney medulla needs, and only small amounts of that glucose is actually secreted to blood. Of course, there is an exception in cases when fasting is so prolonged and starvation is so long that all glycogen reservoirs are depleted from liver, then this pathway of gluconeogenesis in kidneys becomes dominant, or in cases of metabolic acidosis, which we are going to discuss later on in some other videos. So gluconeogenesis is considered to be a pathway which is reversible to glycolysis, and mostly it is literally reversible, with the reactions which are mostly reversible. And actually gluconeogenesis differ at only 3 points from glycolytic pathway, making it total of 4 reactions which are different comparing to glycolytic pathway. So this figure actually represents only an overview of what we did so far. So glucose can be phosphorylated to glucose 6-phosphate, and then the fate of glucose 6-phosphate can be different. It can be converted to glucose 1-phosphate and involved in the metabolism of glycogen, or it can be further degraded to pyruvate in the process of glycolysis, or pyruvate can serve for the resynthesis of glucose 6-phosphate in the process of gluconeogenesis, which is going to be our target for today. So the last but not the least, so this is our target for today. So if we want to provide some kind of definition of gluconeogenesis, we can say that it's a kind of synthesis or biosynthetic pathway of new glucose molecules from non-carbohydrate precursors. It is of crucial importance for the survival of organism because when organism lacks glucose, it can be provided and resynthesized from some other precursors and some other molecules like lactate, amino acids, and so on. This pathway is involved in the maintenance of the blood glucose homeostasis, and it is so important because using the gluconeogenic pathway, sufficient amounts of glucose are provided for those cells and tissues which cannot synthesize glucose themselves, and those are brain cells and erythrocytes. So during prolonged fasting, we all know that liver glycogen pool reservoir is depleted. So after depletion of glycogen pool, glucose is obtained from precursors. Those precursors are predominantly compounds containing 3 or 4 carbon atoms like lactate, pyruvate, oxaloacetate, alpha-keto acids, amino acids like alanine, a glycerol from TAG degradation, and so on and so on. Notice that in this case, some amino acids which are considered to be ketogenic amino acids and acetyl coenzyme itself are not precursors for gluconeogenesis, so they cannot be involved in the biosynthesis of glucose. But I have to say that those components cannot directly be involved in the biosynthesis of glucose, but something that is not topic for today, acetyl coenzyme A can be converted to some ketone bodies. Ketone bodies can be converted to some of those precursors and be involved in gluconeogenesis. So do ketogenic amino acids. But this is going to be the topic for some other videos when we move on with the metabolism of some other biomolecules. So let's get back to our gluconeogenesis. So out of those 10 reactions in total for glycolytic pathway, there are 10 reactions for gluconeogenesis as well. And out of those 10, 7 reactions are actually totally reversible using the same intermediates, the same enzymes. At 3 points, we have 3 irreversible points which employ other enzymes, and they are totally different reactions which are thermodynamically more favorable than those which were found in glycolytic pathway. So the first one, if you take a look here, so this is the first difference, this is the second point of difference, and this is the third point of difference. So in this first point, pyruvate cannot be reversibly converted to phosphoenolpyruvate by pyruvate kinase like we did in the glycolytic pathway, but rather pyruvate is going to be converted to oxaloacetate, and then oxaloacetate in the reaction number 2 is going to be converted to phosphoenolpyruvate. So actually this first branching point is actually coupled with 2 reactions, enzymatic reaction. Then the pathway from phosphoenolpyruvate to fructose 1,6-D-phosphate goes absolutely reversible, and I'm not going to go into details regarding these intermediates and enzyme because we did it in so many details when we discussed about glycolysis, but I'm going to point this reaction. So second point of branching point is that during glycolysis we had the phosphorylation of fructose 6-phosphate to fructose 1,6-D-phosphate under the catalytic function of phosphofructokinase 1, but this time we are going to have rather hydrolysis of fructose 1,6-D-phosphate to fructose 6-phosphate by fructose 1,6-D-phosphate phosphatase. And finally, the third branching point is when glucose is phosphorylated to glucose 6-phosphate by hexokinase in glycolytic pathway, but this time glucose 6-phosphate is going to be hydrolyzed to glucose by glucose 6-phosphate phosphatase, so in totally another way. We're going to go in a little bit more detail regarding these 3 branching point and points of difference. So the first one is conversion of pyruvate to phosphoenolpyruvate, PEP. which is nothing else but the carboxylation of pyruvate. So pyruvate is carboxylated to oxaloacetate, as you can see from this first reaction. The enzyme which catalyzed this reaction is mitochondrial pyruvate carboxylase, because this first reaction occurs in mitochondrial matrix. As all carboxylases, this one also requires a specific coenzyme, which is biotin, and it's more than coenzyme, it's prosthetic group, so it's covalently attached to the enzyme. So the reaction goes in the manner that carbon dioxide is attached to biotin in the presence of ATP. So you have the formation of enzyme, biotin, carbon dioxide. Then the activated carboxylate group is transferred from the activated biotin to pyruvate to form oxaloacetate, as you can see from the figure. In mitochondria of liver and kidney, we can find this pyruvate carboxylase, but this enzyme is not found in muscle cells, and have that in mind. Also, this enzyme is regulatory enzyme, like pyruvate kinase was in glycolytic pathway, and this enzyme is allosterically activated by acetyl coenzyme A. So regarding the fact that acetyl coenzyme A is neither reactant nor product of this reaction, it is a question why acetyl coenzyme A has such an impact of this on this enzyme. So I'm going to make a short digression and discussion about the effects of acetyl coenzyme A to the activity of this enzyme, despite the fact that I'm going to talk about the regulation of gluconeogenesis later on at the very end of this lecture. So anyway, when the concentration of acetyl coenzyme A is increased in cell, it is a typical signal of a metabolic state which definitely requires enhanced oxaloacetate synthesis. Why? Because the overproduction of or over accumulation of acetyl coenzyme A actually drowns the Krebs cycle, so it cannot convert more. So acetyl coenzyme A has to do something to stop it, so it's a signal to enhance the oxaloacetate synthesis, because it has to fulfill the intermediates of the Krebs cycle, which is rotating with a crazy rate when acetyl coenzyme A is increased. Anyway, on the other hand, during fasting, oxaloacetate is converted to glucose in gluconeogenesis pathway. So actually the biosynthesis of oxaloacetate is a kind of anaplerotic reaction. So whenever you need an enhanced activity of Krebs cycle, and in case when there is a lot of acetyl coenzyme A to be degraded, or carboxylation of pyruvate can lead the filling up of the Krebs intermediates. Totally opposite situation is when the concentration of acetyl coenzyme A is decreased. So it's gonna inactivate the pyruvate carboxylase activity. So pyruvate, instead of conversion to oxaloacetate, is gonna primarily be converted to acetyl coenzyme A and further on to Krebs cycle. In case the Krebs cycle is inhibited because of the high levels of ATP or NADH, we have already discussed about this as a marker, certain signals of energy sufficiency, oxaloacetate goes to gluconeogenesis. But we have a kind of problem. Of course, it's not the problem that cannot be overcome, but it's still a kind of problem. And it's about the localization of those processes regarding gluconeogenesis. So carboxylation of pyruvate occurs in mitochondria. So pyruvate carboxylase is mitochondrial enzyme. The second enzyme which is involved in this first digression and difference in gluconeogenic pathway is phosphoenolpyruvate carboxykinase. And this enzyme is found in both mitochondrial matrix and cytosol. All other reactions and enzymes of gluconeogenic pathway are located completely in cytosol. So, either phosphoenolpyruvate or oxaloacetate has to be transported from mitochondria to cytosol to continue the gluconeogenic pathway. So in mitochondria, oxaloacetate can be converted to phosphoenolpyruvate by the action of mitochondrial phosphoenolpyruvate kinase, like we saw in the previous figure. And once phosphoenolpyruvate is synthesized in mitochondrial matrix, it can pass the mitochondrial membrane to cytosol without any difficulties. But oxaloacetate cannot pass mitochondrial membrane. So we have to overcome this problem. So in case we want to transport oxaloacetate from mitochondrial matrix to cytosol, we can do that by the usage of two transport shuttle system. The first one is aspartate shuttle system. It converts oxaloacetate to aspartate in one transaminase reaction, and aspartate AST, aspartate transaminase, is the enzyme which catalyzes this conversion. Aspartate can, as you can see from the slide, aspartate can pass the mitochondrial membrane from matrix to cytosol, and the same reaction, AST catalyzed reaction, converts aspartate back to oxaloacetate, and oxaloacetate can continue the gluconeogenic pathway. So this is the one possibility, so oxaloacetate to aspartate and aspartate back to oxaloacetate by AST. The other is malate shuttle system. And it actually means the reduction of oxaloacetate to L-malate by mitochondrial malate dehydrogenase, which is actually the enzyme of the Krebs cycle. So oxaloacetate is reduced to malate by mitochondrial MDH. Malate can pass the inner mitochondrial membrane to cytosol, and then by the action of cytosolic malate dehydrogenase, remember we mentioned the existence of those two types of MDH when we discussed about Krebs cycle, it can be converted back to oxaloacetate, and oxaloacetate in cytosol can continue its pathway in gluconeogenesis. So this is also one nice figure representing, and it's very informative to give an illustration what we did so far. So a pyruvate can be synthesized from its precursors, lactate or alanine, and pyruvate can then enter mitochondrial matrix. Of course, there are two possible fates in this case of pyruvate. Of course, there are more, but these are important. So pyruvate can be further catabolized to acetyl-CoA enzyme A, but if you have a large influx of fatty acids to mitochondria and increased or enhanced fatty acid oxidation, beta oxidation, you're going to have an accumulation of NADH coenzyme, and if you remember, it directly inhibits pyruvate dehydrogenase, and acetyl coenzyme A does the same. So NADH is going to inhibit pyruvate dehydrogenase, which is going to provide sufficient concentration of pyruvate, which is going to be converted to oxaloacetate. We have already discussed about this reaction. Pyruvate is carboxylated by pyruvate carboxylase to oxaloacetate in a way that carbon dioxide is first attached to biotin and ATP is used for the process, and then CO2 is transferred from activated biotin to oxaloacetate. Once oxaloacetate is synthesized, it can be either reduced to malate or converted to aspartate. Both molecules can pass this mitochondrial membrane to cytosol and undergo reversible pathways to give oxaloacetate again. Oxaloacetate then undergoes reaction number 2 of the first part of the pathway, so by decarboxylation is converted to phosphoenolpyruvate by the action of phosphoenolpyruvate decarboxylase, and then phosphoenolpyruvate continues its gluconeogenic pathway. So the second point the second reaction of this first point is conversion of oxaloacetate to phosphoenolpyruvate, and we mentioned already that the enzyme catalyzing this decarboxylation and subsequent phosphorylation is PEP carboxygenase, and we need an energy from 1 molecule of GTB for completion of this step. Also, we have the release of 1 carbon dioxide from oxaloacetate. So we had an involvement of 1 carbon dioxide from bicarbonate ion, and then we get rid of 1 carbon dioxide from oxaloacetate. So it's not a 0 carbon dioxide. So this is more energetically more favorable pathway of synthesizing phosphoenolpyruvate rather than one which we get from PEP to pyruvate. And from this point on, we have a sequence of several reversible reactions, all up to fructose 1,6-diphosphate. So the third reaction, or the second point, which is specific to gluconeogenesis, is hydrolysis or dephosphorylation of fructose 1,6-diphosphate to fructose 6-phosphate. And again, this is accomplished in energetically more favorable way than those for glycolytic pathway. So the reaction is exergonic and it's irreversible, of course, like in glycolytic. And notice that this time we do not have the synthesis of ATP molecule, because in glycolysis this phosphorylation was ATP requiring process. So one could think of that the reversible pathway will release ATP, but no, this time no formation of ATP. And like in glycolytic pathway, the enzyme phosphofructokinase 1 was the main regulatory enzyme. In this case, enzyme fructose 1,6-diphosphate phosphatase is the main rate limiting enzyme of the metabolic pathway. The last difference in gluconeogenic pathway comparing to glycolysis is hydrolysis of glucose 6-phosphate to free glucose, and the enzyme is glucose 6-phosphate phosphatase and not hexokinase, if you remember. And this enzyme is rather specific because it's membrane bound and membrane attached enzyme. And again, we have the energetically more favorable way of hydrolyzing glucose molecule rather than the pathway which was used for phosphorylation of glucose by hexokinase. Reaction is because of that exergonic and irreversible, and as a result we have the release of inorganic phosphate, and again we do not have the synthesis of ATP molecule because we had a usage of 1 ATP in glycolysis. Again, 101st time, glucose 6-phosphate phosphatase is found only in liver and kidney and not in muscle cells. So glucose 6-phosphate from either gluconeogenesis or glycolysis or glycogenolysis in muscles cannot provide blood glucose. It is reserved for liver only. And as you used to for all metabolic pathway, when we elucidate all the steps of the pathway, we have to calculate the energy yield of the pathway. Of course, these are going to be energy requiring processes and steps, so we're going to use certain amount of ATPs and GTPs for the process. So let's summarize. In the reaction of conversion of pyruvate to phosphoenolpyruvate, we needed 1 ATP and 1 GTP. Then in the reaction that we haven't mentioned at this very moment, but you know it from the glycolytic pathway because it's totally reversible, it's phosphorylation of 3-phosphoglycerate to 1,3-diphosphoglycerate, and for that we need 1 molecule of ATP. And the third one is also mentioned in the glycolytic pathway, is absolutely reversible in gluconeogenesis. It's the reduction of 1,3-diphosphoglycerate, for which we need 1 NADH, which was produced in glycolytic pathway. So if you want to write the total reaction, we have conversion of 2 pyruvates and 2 NADHs and 4 ATPs and 2 GTPs to glucose molecules. So to conclude, the synthesis of 1 molecule of glucose from 2 molecules of pyruvate in gluconeogenic pathway requires usage of 6 high energy molecules like 4 ATPs and 2 GTPs and the presence of 2 molecules of NADHs. Notice that this time we're not calculating NADH coenzymes in energy yield because they're not involved in ETC this time. Which are the precursors for gluconeogenesis? I have already mentioned some molecules, but let's summarize it here. First, lactate, and you can see that lactate can be converted to pyruvate by the very well-known lactate dehydrogenase reaction, and it's also marked and explained by Cori cycle. Especially this pathway is pronounced in anaerobic glycolysis when it's enhanced, for example, in exercising muscles or in erythrocytes which use only anaerobic glycolysis for getting and obtaining energy, or adipocytes after the meal, and so on. The second pathway is conversion of alanine to pyruvate by alanine aminotransferase, and some other amino acids can do the same, which are called glucogenic amino acid, but mostly alanine undergoes this transformation because its keto acid is actually pyruvate. It's described in details in alanine cycle, and we're gonna take a moment analyzing this. But before that, let's mark the third precursor, which is glycerol obtained from degraded adipose triacyl glycerols. So in liver, glycerol can be phosphorylated at position number 3 by glycerol kinase and ATP to form glycerol 3-phosphate. And then glycerol 3-phosphate can be oxidized by glycerol 3-phosphate dehydrogenase to dihydroxyacetone phosphate, which can enter the glycolytic pathway or which can enter the gluconeogenic pathway. So speaking of Cori cycle, this is an explanation. So glucose in muscles obtained, for example, from glycogen or from blood can be involved in the glycolysis to give pyruvate and ATP for muscle contraction. In anaerobic conditions, certain amount of lactate is formed. Lactate is secreted to blood and by blood carried out to the liver. In the liver, lactate first undergoes lactate dehydrogenase reaction to form pyruvate, and then pyruvate can be involved in gluconeogenic pathway to synthesize glucose by the usage of 6 ATPs. Glucose can be secreted back to blood and brought back to the muscle, where it can be used either for getting ATP or for introducing in the molecule of glycogen. Speaking of alanine cycle, here we have a muscle cell and liver cell. So glucose is used for obtaining energy for muscles, and we get pyruvate. And pyruvate can be converted to a lactate in anaerobic conditions, but in cases of muscle protein breakdown, we get certain amount of alanine, which can be also formed. So pyruvate can be converted to alanine by alanine aminotransferase activity. And then alanine is secreted to blood and brought to liver. In liver cells, alanine again undergoes transaminase reaction by ALT catalytic function to form pyruvate. In this transamination reaction, this amino group, which was caught from the muscle protein amino acid catabolism, is then released in the form of glutamate. Glutamate undergoes deamination to release ammonia, and ammonia enters the urea cycle, which is unique for liver cells. On the other side, pyruvate can enter gluconeogenesis to provide certain amounts of glucose, which can be brought back again to muscle cells and obtain and start the new cycle and new ATP synthesis. Speaking of the regulation of gluconeogenesis, regulatory enzymes are the ones which are at the same points where we had regulatory enzymes for a glycolytic pathway. So in glycolysis, we had caxokinase as the first regulatory enzyme. So in gluconeogenesis, it's going to be glucose 6-phosphate phosphatase. In glycolytic pathway, the second and main regulatory enzyme was phosphofructokinase 1. At the same point, the main regulatory enzyme of gluconeogenesis will be fructose 1-D-phosphate phosphatase. And finally, the third and the last regulatory enzyme of glycolytic pathway was pyruvate kinase. At the same point, pyruvate carboxylase and PEEP carboxykinase are going to be regulatory enzyme for gluconeogenesis. Speaking about the regulatory mechanisms, we have two ways. Like for all other metabolic pathways. Some of those we have discussed before. It can be allosteric regulation. Some of the allosteric activators and inhibitors we have already discussed by the effects of acetyl-CoA enzyme A to pyruvate carboxylase. The other way is covalent modification, which is CAMP driven or directed, or CAMP dependent covalent modification, which means phosphorylation and dephosphorylation, which is also promoted or inhibited by the action of hormones. We have discussed about detailed mechanism of those pathways when we discussed the regulation of glycogen metabolism, and it's almost the same. One of those points of regulation is the availability of substrate. So it is very important for the regulation of this pathway, because if you don't have precursors available, regulation mechanisms are of less importance. So predominantly it is about the glucogenic amino acids, and predominantly alanine, and to a lesser extent lactate, which are precursors for gluconeogenesis. Then we have allosteric regulation of gluconeogenesis, and we have already mentioned the effects of acetyl-CoA enzyme A, which I emphasized here, and its action on activating pyruvate carboxylase. But here I have compared glycolytic and gluconeogenic enzymes. So phosphofructokinase 1, which is a glycolytic enzyme, if you remember, was activated allosterically by ATP and citrate, and inhibited and activated by the lack of energy in the form of AMP signal, and fructose 2,6-D-phosphate, which is a very specific molecule. And in gluconeogenesis, fructose 2,6-D-phosphate actually inhibits fructose 1,6-D-phosphatase, and AMP does the same. So in glycolytic pathway, pyruvate kinase is inhibited by alanine and activated by fructose 1,6-D-phosphate. The same enzyme, the enzyme in gluconeogenic pathway is activated by acetyl-CoA enzyme A. Phosphofructokinase 2 and fructose D-phosphatase 2 are enzymes involved in the formation, phosphorylation and dephosphorylation of fructose 2,6-D-phosphate. So they are also driven allosterically by different compounds, which are marked here. And to conclude, the hormonal regulation of gluconeogenesis is accomplished in the same, almost the same way as we had for the metabolism of glycogen. So I'm not going to repeat the explanation of the same mechanism, but the main hormone which regulates gluconeogenesis is glucagon. So it decreases the level of previously mentioned fructose 2,6-D-phosphate. It inactivates pyruvate kinase. So less phosphoenolpyruvate is converted to pyruvate, and it activates PEEP carboxykinase during fasting periods. Catecholamines like adrenaline or epinephrine, they act in the same way as glucagon, using the same mechanisms, and those are the same mechanisms which we explained by the terms of glycogen metabolism. So anyway, this was all regarding gluconeogenesis, and it was some kind of the state-of-the-art review of the overall metabolism of glucose. So this was the last topic regarding the metabolism of carbohydrates. The upcoming videos are going to be dedicated to some other biomolecules and some other ways of regulating their metabolism, catabolism and anabolism. So at the very end, I am reminding you to feel free to contact me and to provide me some suggestions. Which topics would you like to hear? Whether you need some further additional explanation regarding the topics that we have already done, and so on and so on. So anyway, this was all, and until some other topics, some other biomolecules, please stay safe, stay healthy, be smart, be pretty, and definitely bye bye and have a nice weekend.